But even with the noise files right now and the paired germline sample, we are looking into a lot of specifics and detailed variants and one by one.
Hmm. Yeah, no, that makes sense. So, as you can notice, I just started the recording and I apologize for that. So just to recap for the recording, we're having an open discussion on whole genome sequencing and how to report that, and also what's the best way to implement that within our laboratories and different settings. And so the first question I had posed was how many people have actually dealt with whole genome sequencing data. We had one in our and then the second question, which is up on the screen right now, is should a paired normal tissue be required for testing? And so it's either or preferred, but the point being that without the and that you end up with a lot of additional variants in your list that require attention and manual effort. Oh no. So, any other thoughts on this? Hi, Peter.
Morning. So, back in 2016, my group published two papers, but basically our approach wasn't whole genome sequencing, but whole gene sequencing. So the papers were about patients who had probably inherited breast cancer, but never had no mutation had ever been found in those patients. So those represented a, a, a cohort that we could justify whole gene sequencing on. And we did find, of course, some variants that were missed by conventional exome sequencing that were actually in the exons. So one of the advantages of whole genome sequencing is you get better coverage towards the ends of exons than you do from exome sequencing. And so, even conventional sequencing benefits from it. But the problem that is, the papers were, it was a challenging to decide how to position in many cases when we found variants that we thought were pathogenic to confirm them experimentally, right? So, if my lab's expertise, uses information theory, and we analyzed splicing mutations and transcription factor binding site mutations, and we could say these variants can be prioritized, but without the experimental evidence that increasingly ACMG, AMP, and even ClinVar are now pushing for. You can't go beyond the concept of prioritization. So really, while we're talking about issues like coverage and paired normal tissues and so on, at the end of the day, there are limits to the interpretation of whole genome sequencing or whole gene sequencing, even in situations where clinically you have a mandate to do it.
That's a really good point. Um, yeah, so you have pluses and minuses to every test, it seems. For whole genome, a lot of times we think of just the cost, not necessarily the technical limitations. So, no, I really appreciate your perspective there. All right, so
It's interpretation. It's how far can you go to interpret, and can you actually convince a clinician to use the other bioinformatic technique to actually counsel their patient with this information? I think the answer is no, you can't. There's another issue too, but I'll save that for later in the discussion regarding interpretation.
Sounds good. All right. Manuela, go ahead.
Yeah, maybe a couple of thoughts. I completely agree that it's difficult to interpret most of the variants you would cover with whole genome sequencing. However, there are a few things which you would find or could be are able to analyze which you're not able to do with a targeted panel. I mean, of course, you can have panel sequencing with a CNV backbone, for example, which is one thing, which is already a problem. But then you also get I mean, they're also more expensive from that point of view. Um, but what we quite often see is that we have like intragenic structure variants which do affect um, also the genes, and they do have a consequence which you wouldn't see neither with whole exome sequencing nor with a panel in sequencing as such. And the other point, I mean, that's with respect to the patient perspective. But I see a really important point in building up these kind of cohorts where you have real world evidence and discussion of patients in molecular tumor boards. And you can basically learn from those for the next patients. And I think that's a major advantage of using whole genome sequencing. But I wouldn't stop there, actually. I wouldn't stop at the DNA level.
So with those extragenic findings, do you find that there are more of those in like hematologic malignancies versus solid tumor? Or are they pretty ubiquitous?
Okay, I have to admit we don't have hematological tumors in whole genome sequencing. We didn't go there yet. So as of now, we only have solid tumors, where it's quite easy also to do the normal pairing for
Mhm.
for the analysis. So I can't comment on that.
Sounds good. And what do you use for the paired normal? Do you use adjacent normal tissue or blood, or what do you use?
We use blood mostly as it's the most easiest one because I mean that's also a question of procedures. A lot of patients are coming from different cities or like outside of Berlin as such. So their samples are usually FFPE material. So it's hard to have additional adjacent material or good enough adjacent material.
Sounds good. Thank you so much. So I am going to give you another poll question here. I have five prepared, and they're working brilliantly to advance our discussion. So I love that. Here we go. So in the chat now, you'll see a new link, and that is for the next poll question. And then once responses start rolling in, I'll go ahead and share my screen again, and we can prompt a little bit more discussion. And so we've already touched a little bit on this question when Peter was talking about techniques being used for whole genome sequencing or whole gene sequencing, but this will help us to further that discussion, I hope. All right, so I'll go ahead and share my screen. There we go. So as you can see here, somebody very cleverly wrote short read NGS plus RNA sequencing, which I think a lot of the clinical labs move in that direction toward like a whole genome, whole transcriptome, or large panels for one or the other. But yeah, any thoughts on this? I'm a little surprised that nobody answered long read, but yeah, it was fun. Peter, I see your hand is raised.
So I guess I'll talk about what we did in our two papers back in 2016. So we basically designed oligos to capture whole genes, but we skipped the um, transposable elements, the ALUs and the L1s. So it was basically a masked capture. And we did get, 20, 30 fold coverage for many of the sequences. There were some that were a little lower, but when we had a potential candidate variant, we then went in and sequenced that by resequenced it by Sanger. But it worked. And of course, it's much cheaper to do this, and it works. I mean, you can make libraries and so short read sequencing does have the additional cost of having a capture pool that includes intergenic and presumably upstream and downstream sequences. That's what we did, and it works. So there's some additional cost from those pools, but at the end of the day, the sequencing itself is conventional. The challenge, in my view, is the interpretation.
Any other thoughts?
So would any of you like me to put those citations in the chat in case
I
you're
would love
interested in
that.
reading
And
them?
then if you're okay with it, I'll add those links. I'll add those
Okay, I'll
links
do
to
that.
the video. Yep. Thank you. I
Okay,
appreciate that.
let me
Maybe a
go get
quick
it.
thought on long read sequencing from what we have seen. So, I mean, you have two options now with ONT being fast and quick and quite cheap for diagnostic because you also have methylation there. But it's very, very shallow, so it's hard to really go into variants. But same with PacBio, which is very accurate. But I think one lane gives you 30x coverage for one patient, and it runs for 48 hours. So this is a time question, not only a money question. I mean, uh, you would have to get really sufficient coverage for small variants. This is just not feasible, in my opinion, as of now. Especially if you then again want to have a normal as a comparison. Um, yeah, making it really difficult to use long read sequencing.
That makes sense. I know that a lot of times we really want to be able to capture like our cis trans variants that are in the same gene. Um, and a lot of times we can do that with short read depending on how close those variants are together. But long read sequencing, I agree, is more emerging, but to me it's exciting from the perspective of structural variants and things of that nature. Um, all right. Well,
Just
thanks.
this,
And any other thoughts? Otherwise, we can move on to our next full question, which I think will be an interesting one and help drive some discussion on the types of variants we want to be calling from whole genome sequencing. Um, because I think a lot of people see the potential in whole genome sequencing and see all the different types of data that can come out of that and, want to report everything we can. And I'd like to hear perspectives on what people think about, these different types of data. So take your time with this one. It's a little bit longer. Um, I'll do that. And I see that first link, Peter. Thank you so much. All right. So take your time with this one. It has a number of different checkboxes with different types of data that can be, you know, derived from a whole genome.
May I ask a question? Should be reported? Is that in a sense of like has to be versus can be?
I think like ideally, this is meant to be like an idealistic survey, not necessarily like a, I mean, not necessarily like what are you doing in practice today, but
Mm.
like in your mind, what would you like to see?
It's an interesting question because I think there are some of these things that could be easily reported with a lot less effort from other assays. So maybe, maybe you could get it from whole genome, but you don't need to. So
Yeah.
it's tricky. I don't know how to answer the question.
And that's exactly kind of my thought when I was putting this together too, was like it's it's interesting because you can report this information, but I mean, do you want to take all of this into a whole genome versus something else? And you never know, like from a clinical lab standpoint, you never know if somebody's going to order a whole genome or order, but usually I'm guessing they'll order other ancillary testing leading up to a whole genome. It's usually how the clinical workflows work. Um, Kind of the low-hanging fruit before you get the big test. Um, all right, so we have a number of responses. So I'm going to go ahead and share my screen again.
You're
Or
sharing.
I am. Oh, great. Cool. Great. I never stopped the last time. Um, so here we go. So we can see most people say small variants, copy number variants at a gene level, tumor mutational burden, microsatellite instability. Um, So that's 6 out of 6 people. I feel like those are somewhat like common with kind of any large panel. That's what we're getting from the large panels now. Let's see. And then also scoring high are potential fusions or structural variants based on a DNA footprint. So like if you have, I'm thinking like Kia 1549 BRAF and a pilocytic astrocytoma, you would probably point that out. LOH, or loss of heterozygosity, also scored high. That's cool. And then potential germline variants related to cancer. So calling out um, variants that are potentially germline and related to a patient's phenotype. Um, Interesting. Then copy number variants at a chromosome level is an interesting one too. Um, so that one also scored high. So out of 7 responses, so if we would take like um, majority rules here, 4 and above, we're reporting everything off of this list except for chip and mutational signatures, which is a crazy amount of data, but so interesting. Um, Anybody want to comment on anything that kind of struck you as interesting when you're responding to this question?
I mean, from my perspective, I think that I'm not doing any of this, so I'm just looking at the downstream data. So I'm 3 steps removed probably, but thinking about what you can get from a whole genome and what you can get from smaller panels, and I know that there are it's a competing question, right? Whether you want to do a whole genome, which is a lower coverage and much more expensive, but you can do one assay and get everything. So from that perspective, like you can, bioinformatically restrict and get your small nucleotide variants and your, gene level copy number variants. I think that things like microsatellite instability, you can run a quick PCR test and get the same information that you don't need to extract it. And I don't know how hard that is to extract from whole genome. Maybe it's really easy. Like this is where the limit of my understanding of this technology. But I think I would focus on things like loss of heterozygosity. And I think copy number at the chromosome level is something that's much easier to get from a whole genome than from really targeted um, assays. Um, genomic complexity, questions of chromothripsis, and the bigger questions, I think, are where it really is valuable. I don't know. Those are my thoughts.
I like it. I mean, my background is cytogenetics originally, so I like hearing people say that chromosome copy number is something that we could be getting from whole genome, and we should be getting from whole genome. Um, and things like chromothripsis, we may not know exactly what that means as far as clinical relevance, but it's something that we can absolutely get from the data. Um, so it's really interesting stuff. So thank you for sharing.
Yeah. I mean, I think something like minimum residual disease is something that you really could do from a much smaller assay where you've already identified some key variants and you can just look for those. You don't need to do a whole genome to track minimum residual disease.
Right. Right. Any other thoughts? Manuela, I see your hand is up.
Yeah, I think, I mean, again, one would have to ask the question whether it's for the patient and at that moment, or whether we are asking bigger questions also for cohorts. And I think that's a
Yeah.
major point. What I do find interesting in the list Beth put up here is mutated biological pathways, because often variants are discussed one by one and like just in maybe a connection to two or three others, whether there's any resistance mechanism. But with whole genome sequencing, we don't just only have a focused view on the most relevant genes, but we do have, hopefully at some point, an overview of really pathway changes. And we might get a more holistic view. So not that I would say that it's really relevant as of now for patients, but I think it will be become more and more important long term.
Interesting. Yeah. No, I think that's pretty cool. Um, Peter, go ahead.
In light of Manuela's recent comment a moment ago, I want to make a comment about non-coding variants. So again, this is something we've studied for quite a few years, and so a single variant can have multiplex effects, or in fact, a single variant cannot have any effects, even though it should. So I'll start with the second one. We published an NAR paper where we took data from the ENCODE project and made information theory models for over 120 transcription factors, and then scanned genomes for mutations in those binding sites. So not the factors themselves, but the sequences they recognize. And we ended up going back to an earlier study that we had done. What we found, and what's been found by others, including at EMBL, is that for transcription factor binding sites, they cluster in promoters. So there's a lot of redundancy. You will find a cluster of multiple sites, and very often, I mean, in some cases, these become enhancers, multiple binding sites of the same type. So you can have a mutation that abolishes binding at one of the positions, at one of the sites, but yet none of the others are actually affected. So we proposed that something called information density, so that if a transcription factor is looking for its target and it has multiple locations that it can bind to regulate a gene, if it has multiple locations, then a mutation in an individual location is buffered by the others, right? And so as a consequence, you have evolution selecting for basically this redundancy so that a critical regulatory effect of a particular transcription factor isn't abolished by a single point mutation in a promoter. And in fact, promoters are actually very, very polymorphic. And this is absolutely essential in order to preserve the regulatory networks that we have in our genomes. So the first point is a single point mutation that looks like it abolishes binding of a transcription factor itself may not actually have any impact on the expression of that gene. It takes multiple changes to actually abolish that regulation. The second thing we noticed is just the opposite when it comes to splicing. So we can have a mutation, not necessarily even in a splice site. It can be somewhere upstream or downstream from the natural splice site, but activates a cryptic splice site. Sometimes the mutation in the natural splice site is leaky. It doesn't abolish splicing, it just weakens it, and hence you get lower amounts of expression. The other thing that happens is that sometimes weakening a natural site will activate a neighboring cryptic splice site. So what I'm getting at here is the ACMG AMP criteria need to be revised because the outcomes, both the inputs and the outputs of single nucleotide changes cannot be basically described in terms of those criteria. If in fact there are going to be multiple isoforms generated by a splicing mutation, including intron inclusion, which we don't generally look for in the lab at all because we use PCR primers that flank exons and look for their presence or absence. So if you have a failure to recognize an exon, then it gets included in a very long transcript that has a lot of intronic sequence. And in our analysis of TCGA splicing mutations, we found this happened a lot. So there's a whole class of outcomes that we're generally not looking at using the techniques that are available to us. And whole genome sequencing with transcriptome sequencing will reveal these things, but we simply can't interpret all of these different potential outcomes. I think, so this was the point that I wanted to make is that, and I've talked to Beth about talking about this later in the year. The ClinVar has now got a set of criteria for providing functional evidence. And we did this for all of the TCGA and ICGC genomes and exomes. And we added 500,000 variants that affect splicing to the TCGA database. Now, these are not variants where we know anything about allele frequency. These are variants where we have a predicted effect on splicing that's validated by transcriptome analysis. And I think that that's going to be the way in which we are going to be able to incorporate whole genome sequencing into clinical practice. As more and more multi-assay papers come out that describe ways of providing functional evidence for different kinds of mutations, then clinical labs like yourselves will be able to just simply look up the consequences of mutations in ClinVar. And I think we're going to have to be available to try to explain some of the things that we're reporting to ClinVar. But I think at the end of the day, that's how this gets put into practice.
So I have a question for you, Peter, about when you have kind of those multiple kind of redundant regions that work together to create kind of like a pathogenic mutation collaboratively. Like if you have one area that if you mutate it, nothing happens, but if you abolish the rest of it, it's gone. How would you like what kind of class would you make for that within kind of like an ACMG like structure? Would it be like a partial pathogenic variant, or like, I mean, how would you even describe that? Am I thinking about that correctly?
Yeah, you are.
Okay.
So the thing that's funny is that early molecular biology is full of these examples, right? So when we all learn about promoter biology, we learn about, for example, Pierre Chambon's famous papers where he would do knockouts of promoters by deleting pieces of the promoter to define where they were, where the promoter is and what the critical element, where the critical elements were. And there are thousands of papers like this for almost every gene you can imagine. where they're trying to dissect the promoter by doing deletion mutagenesis. Sometimes it's linker mutagenesis, but the deletion mutagenesis would remove a whole set of binding sites, a cluster of binding sites. And so you would see an effect on expression, whether it was a luciferase assay or some other type of assay, chloramphenicol acetyltransferase, it doesn't matter what, right? You would put this thing on a plasmid, transfect it into a cell, delete that piece. That piece had multiple binding sites in it, and hence you would now start to see a reduction in expression, right? So any sort of mutations that involve deletions of a chunk of DNA from the promoter are going to have a profound effect on the expression of that gene. Now, if that gene plays an important role structurally, regulatory, whatever, you're going to have to look at the regulatory pathways in which it participates, and then ask if that gene wasn't present, if its gene product wasn't present, what other genes would be impacted from it, right? So if it was a transcription factor, well, then there's a whole battery of genes. But if it's some structural factor where you need this gene to, I don't know, express collagen, for example, well, then you might expect to see a collagenopathy because you have now prevented the transcription factor from activating the collagen gene. So I think we're going to have to look at network biology as a way to interpret regulatory effects. We can't simply go through an algorithm that ACMG and AMP provide.
I'm thinking of other algorithms too that might be a little bit more accessible, like what the TP53 database uses as their non-functional, partially functional type of ontology. That might be more applicable to what you're describing, but really interesting and thought-provoking. So I appreciate your input on that. All right. Did we want to talk a little bit about, let's see. Yeah, so pathway biology. What information should we be able to garner from a genome? I feel like when I talk about pathways, usually I'm looking at more of the transcriptome data and looking for relative expression of my RNA, but I wanted to know if there was anybody who had thoughts and ideas about how to look at or interpret those biological pathways. Cheryl, I saw you came off mute.
I was thinking about the previous question also, but I mean, this question, and I agree with you that in terms of activated pathways, I think RNA is a better readout for what's actually happening. But I do think that there's also a role for selection and, synthetic lethal type of things, similar to what Peter was talking about, where you have one mutation that may or may not contribute, but then when you have a mutation in two or more genes that they lead to enough depletion or activation that you activate a cancer process. So I think those kinds of things, we know about some of them with respect to, PARP inhibitor, for example, is a synthetic lethal interaction with DNA damage. And if you have more DNA damage than, or more disruption to certain DNA damage pathways, you're going to have, a synthetic lethal effect where you don't have enough redundancy anymore to be able to accomplish what you need.
Yeah, I think another point is what we looked at is tumor mutational burden. I mean, if you have a high tumor mutational burden, you usually it's a good indicator for using immune checkpoint inhibition as a treatment. But if you have, we found that if you have like a medium tumor mutational burden, around 3 mutations per megabase, you actually see that patients are less, their response to targeted therapy, which is based on usually one variant or one gene, is really inferior if you have this like in between. And I think that's the point where pathway analysis also could come in. It's probably not synthetic lethal as such, but it's resistance mechanisms which you're kind of
Yeah.
having there. And if this is captured in a panel, that's fine because it's well studied, like MAP kinase pathway, PI3 kinase pathway. But maybe it's some resistance mechanism which we don't know yet, and then I think there it's where we can actually learn something from whole genome sequencing, just to understand how those pathways are connected.
I think that's a really good point, and I think it's also been really interesting to see in hypermutated samples in colorectal and particularly in endometrial cancer, we see an incredible concentration of mutations in the PI3 kinase pathway. So multiple PTEN, PIK3R1, PIK3CA mutations, and like, what does that mean? We don't know, but it's very interesting to see how concentrated they are, how many you get in those hypermutated samples.
Almost like a mutational signature in itself.
Yeah, yeah.
Yeah.
Yeah, just
So
to add to those, so in my previous life at Semaphore and now GeneDx, so we had this exact survey with like oncologists and healthcare providers, and there was quite an overwhelming response for the need to have like pictorial representation of these biological pathways on the report, especially with community oncologists. They were like, "Oh, this is going to be extremely useful just to get a bird's eye view of what's happening in the patient without even going into the details too much." But yeah, um,
that makes a lot of sense. Thank you. Thanks, Uma. Um, Dee Meyer is in the chat asking a question about can a mutated biological pathway serve as an actionable biomarker today that a clinical lab worker would report? I would argue yes, it could, because you have those like actionable pathways, so your PI3K mTOR type pathways that are quite active. So I think yes, but I think a lot of those biomarkers are also in and of themselves kind of actionable. So um, yeah. Well, no, thank you. That's a good
I
way to
think it
think about
gets
it.
I think it gets trickier when you're talking about the RNA output. So I think for
Mm,
the mutated, groups of mutated pathways, you group them together on the port and it's, oh, you have a BRCA1, two BRCA2s, and an ATM. Maybe that strengthens the desire to potentially use the PARP inhibitor if you've
use a
hit
PARP.
so many
Hmm.
things in that pathway. But, and I think that's well documented, and you would be probably reporting that same drug for any one of those mutations. Whereas when you're looking at, or, to Manuela's point, if you have, say, an EGFR mutation and also a MET amplification that's known to be resistant, that those two would be reported together. Um, uh, but I think the other question about expression when it's not directly related to known mutants gets a lot trickier because it's not clinically validated.
Hmm. Peter?
Oh, I actually wanted to, I'm sorry, I lost the screen here. Um, I can't, hmm, weird. I've got your original survey. I wanted to respond to Uma Devi's comment about pictorial representations. We were doing some work that I think might be useful. It's not just the pathways themselves, but what you want to show in the pictorial representation is whether the genes are upregulated or downregulated. And so we published a few papers on gene signature and actually have symbols for whether or not we'd color code the proteins in the context of where they are in the cell and whether they were upregulated or downregulated. And I wondered if that's what she's referring to, or when she talks about a pictorial representation, is she referring to something else?
no, that's part of it because at that point we didn't have expression data. So it was mostly mutational data. So we had color coding done for genes that were mutated, but but where we want to be is exactly what you just mentioned.
Okay, so I'm going to put a couple of links in the chat for you to go look up. And
Oh,
there's
mhm.
some figures in these papers which I think you can use. I'm not saying you want to use the papers themselves, but the way that we've described the changes that lead to resistance or sensitivity to chemotherapy drugs may be relevant to what it is you're looking for.
Great. Thanks,
Okay,
Peter. Yeah.
you're welcome.
Fantastic. Thank you. Oh, that's awesome. See, I also wanted to talk about, I saw that only a few people had responded about not minimum residual disease, mutational signatures. I've noticed like when we have high mutational burdens, we may see like very concrete mutational signatures. And I was wondering what, if any, experience this group may have had with looking at those or calling mutational signatures has been probably the more challenging part. When it's a really high tumor mutational burden and you have all C to Ts, that's pretty easy to call based on context. But I'm kind of wondering what all of you think about mutational signatures and the use and the calling of those.
Oh, we haven't tackled mutational signatures, and I think it might be challenging because if you're working from FFPE material, you also have all those transformations and transitions coming from the treatment of the samples as such. I guess there are people who are working on that. Would be really interested in hearing thoughts on that. I assume it will be challenging to do in a clinical practice.
Yeah, I was
Yeah.
gonna, oh, sorry, Beth, go ahead.
Oh, yeah, I was going to say it is pretty challenging because a lot of the mutational signatures tend to be overlapping. And so any comparison that we're doing is like, percent matching to this mutational signature. And it's not always, you don't always have a great match. And so it's interesting. Go ahead, Cheryl. Sorry to interrupt.
I was going to say something very similar. I think there aren't a lot of, or many, or any mutational signatures that are validated for clinical use. I mean, there are a very few, I think. And so you need to divine them, right? You need to figure out like what constitutes a mutational signature. You need a lot of patients to validate that mutational signature and what it means clinically. So I think it's very tricky. It's a you need a lot of data and a lot of computation to be able to do it. And I think that there are labs that are starting to do it with methylation signatures that are having a lot of success categorizing different neuro-oncology uh, subgroups, but I'm not aware really of mutational signatures per se that are being clinically used, and we're certainly not applying anything in our work.
Another thing that we've seen in a lot of these like high tumor mutational burden scenarios is the frequency of indels and frame shifts. So like small deletions, insertions um, is higher in a lot of those. And there have been some studies that have come out that have suggested that those patients actually respond better to immunotherapy. So that's been an interesting thing that I haven't had a chance to study, but I've seen it in practice, and it's pretty interesting stuff. So yeah, we're seeing. Okay, great. So we have another paper from Peter et al. So that's great. Thank you so much. All right, cool. So I have one more question, and this has been a wonderful discussion. I'm really pleased that all of you have joined today and have been so great with participating. Um, So the last question I have, I will go ahead and share this. There's the responder link. I'm putting it in the chat right now. And it has to do with should the data reported from whole genome sequence vary by tumor type? As we've seen in some commercial reports, you might see LOH, but only for ovarian tumors, or like a HRD, but only for, ovarian or breast and ovarian. So wondering, do we think that the data should vary based on the type of tumor that we're looking at? And once I get some responses, I'll go ahead and share my screen again. I love it. We got it. It depends. That's perfect.
I chose, but I would have said it depends.
Yeah. No, I'm like, oh, yeah, I
No?
wasn't even sure how I would
It
answer
depends
this.
is like my favorite answer to everything.
Right?
Yeah, that was me as depends at least.
So
But
what
I
are
mean,
the,
the thought is like, who do you put the work on? Is it the clinician, like making sure that he sees all variants which might be of interest, or is it at the level of molecular pathology and already preparing beforehand? And I think it really depends on who you are talking to and on what level, like who are you handing out reports? How skilled are the clinicians you're working with? How much can they interpret the results? And I think that's the point why I said
that's the
it
classic
depends.
reporting question, like who's your
Yeah,
audience? How,
exactly. So I don't know, Cheryl,
Yeah.
why would have you said it depends?
Um, I was thinking it depends on the tumor type, that there are certain things that are going to be important in some tumor types and less in others. So I guess it is important that you know what tumor type you're looking at. I think there are certain genes that are really important in heme malignancies, for example, where they really don't have any bearing in a solid tumor and are most likely to be chip in a solid tumor, but could be a driver in a heme tumor. And I think that there's probably a lot more like that than we know, and the relevance of certain mutations, depending on the tumor type, is going to be important. And the more data you have, the more, as you said about the audience, you need to be careful about what kind of information you're giving, depending on who is reading the report.
thank you. That's really helpful. I see that we have 2 minutes left. So if there are any other kind of uh, revelations that are coming to mind, feel free to share them now. But otherwise, I just wanted to say thank you all so much for contributing to this conversation. It's been really educational for me and great to collect these perspectives of all of you experts in our field. So really excited. Yes, Peter.
So I guess my final point is that, the first question you should ask is WGS worth doing? And the answer is, of course, yes. And the reason is, even if we don't know now how to interpret all the variants that come out of WGS, we will know someday, right? And with that genome in a patient's chart, then, as we learn more about it, if in fact there's some mechanism for revisiting the chart, I think, patients want answers, right? I mean, that's the reason why you guys do what you do. They want to know, uh, what is genome sequencing going to do for me, right? And at the end of the day, we have to be willing to say, look, we understand some things, but we don't understand everything yet. And, you should stay in touch with us. I mean, that's when I was running a molecular diagnostics lab, I would get recontacted by patients all the time. I don't know whether or not that's possible under the current regime, but it certainly is, as you become available, as more resources become available to you guys who are actually analyzing genomes and exomes, and your work becomes more efficient, then it will be possible for you to respond to those secondary inquiries.
Mm-hmm. Absolutely. And yeah, I've always been a believer that, clinical implementation often precedes exception of, clinical utility and full understanding. So it's good to kind of hear that perspective reiterating my own thoughts. So thank you for that. Anyhow, we are at time, so thank you all so much for joining today. This will be posted on the VICC YouTube channel, and I will add all of the links and papers and stuff to the comments on the recording. So thank you so much for joining, and we'll see you all next time.
Thank you.
Take
Thank
care.
you.
Bye-bye.